Hello, my name's Mervyn Singer. I'm at University College London in the UK. I'd like to talk to you today about mitochondrial physiology, a wonderful subject. I've got no conflicts of interest to declare, but what I would hopefully emphasize is that this is a complex topic, but I'll try and make it as simple as I can. The mitochondria are wonderful organelles and my favorite organelle. They're present in almost all cell types in the body. Apart from making ATP, they're a major provider of body heat. They use most of the oxygen that the body utilizes. They're the major source of free radicals in the body, a major target of nitric oxide. They have a crucial role in triggering cell death, in regulating intracellular calcium. Not only are they important for making hormones, but they're a site of action of hormones. So thyroid function is dictated through its modulation of mitochondrial activity. They have a major role in producing vitamin D, in lipid metabolism, and immune function. They likely came from probacteria millions of years ago, and these were incorporated into prokaryotes to make eukaryotes and generate sufficient ATP to provide the energy to allow us to become the sophisticated organisms we are today. So this is a schema of the anatomy of a mitochondrion. There are outer and inner mitochondrial membranes, and the inner mitochondrial membrane is folded many times into cristae, which increases its surface area. Inside the cristae is the matrix, which contains lots of proteins, enzymes, and so forth. But the enzymes involved in the electron transport chain that generate ATP are actually sat on the inner mitochondrial membrane, as is ATP synthase that makes ATP from ADP. The mitochondria has its own DNA, which makes gene products for 37 genes. However, most of the mitochondrial protein actually comes from nuclear encoded DNA. So there's a combination of nuclear DNA and mitochondrial DNA that make up the total mitochondrion. This schema here shows some of the biochemistry. I'll talk through it simply, but hopefully in a clear manner. The cytosol is shown in blue on the outside, and that's where glycolysis occurs. So glucose is metabolized to pyruvate, which enters the mitochondrion. The pyruvate that's not used goes to lactate, and the lactate can be returned back to pyruvate if it's needed as an energy substrate. The pyruvate feeds in via pyruvate dehydrogenase to acetyl-CoA into the Krebs cycle. Amino acid alanine can also be used as a fuel source, and that feeds into pyruvate, as can fats. So short-chain fatty acids can come in to the mitochondrion directly, whereas medium and long-chain fatty acids have to be converted to carnitine and then reconverted back to fatty acyl-CoA, which then undergoes beta oxidation. And the beta oxidation can go to acetyl-CoA and then into the Krebs cycle, or down straight into the electron transport chain via coenzyme Q. Succinate can also be fed in directly into the Krebs cycle. So reducing equivalents from the Krebs cycle, NADH, FADH2, donate electrons to the electron transport chain, to complex 1 and complex 2 respectively. And there are 4 enzyme complexes in the chain and 2 carriers, coenzyme Q and cytochrome C. Electrons pass down the chain from complexes 1 and 2 to ubiquinone, coenzyme Q, and then on to complex 3. They're then carried, the electrons, from cytochrome C to complex 4. Another name for complex 4 is cytochrome oxidase. And that's when most of the body's oxygen is used, on that one enzyme. Oxygen's the terminal electron acceptor. As the electrons pass down the chain, hydrogen, protons, are transported across the inner mitochondrial membrane, and that creates a gradient which powers ATP synthase to to make ATP from ADP. And then the ATP is transported out of the mitochondrion to the cytosol via a translocase, ANT, and ADP can return in its place to be regenerated back into ATP. Oxygen can also be used at coenzyme Q and complex 1 to generate superoxide. So a small amount of our oxygen utilization in health goes to make free radical species, which have important roles in cell signaling. In disease processes, this can increase greatly and be pathological. Protons don't necessarily need to cross via ATP synthase back into the mitochondria. They can pass through the membrane through uncoupling proteins, and as a consequence, the energy is dissipated as heat. And as I mentioned before, much of the body heat arises from this process. 2 molecules of ATP are made by a mole of glucose with glycolysis, 2 from the Krebs cycle, and 24 to 30 from oxidative phosphorylation. So you can see the importance of the electron transport chain. Peter Rich, who's a biochemist, put out this very nice paper in Nature, again highlighting that most oxygen is used by cytochrome oxidase, and a huge amount is present in the body to support oxygen consumption, 10 to the 19 moles. And if you opened up the surface of the inner mitochondrial membrane, this would cover 14,000 square meters, he estimated, 50 tennis courts in square meterage. There's a huge flux of protons across the inner mitochondrial membrane, 3 times 10 to the power 21 per second, and that generates a huge turnover of ATP, 9 times 10 to the 20 moles per second. So there's a turnover rate of ATP equivalent to our body weight. Truly phenomenal. And that's enough to power a 100-watt light bulb. Most cells require oxygen to function, albeit to varying degrees. So hepatocytes, cardiomyocytes are full of mitochondria. Neutrophils have relatively few, but rely more on glycolysis. And only red blood cells in the whole body lack mitochondria completely and are totally reliant on glycolysis. Let's talk quickly about oxygen. So there's this gradient of oxygen from the blood through the capillaries, through the interstitium, the cell, and into the mitochondria. And the level there is really low, about 0.1 to 1 kilopascals compared to 13 in the blood. So the oxygen is carried by convection to the tissues and then diffuses across the different barriers to get to the mitochondrion. As I mentioned before, this is a very, very low oxygen tension in the mitochondrion, but fortunately, cytochrome oxidase can operate at very low oxygen concentrations. This is in isolated rat mitochondria, and you can see that the activity of the enzyme only starts dropping when the partial pressure is below 0.1 kilopascals, so very, very low. How's the oxygen used? Most for ATP synthesis. This is using the liver as an example to make protein, sodium pumps and other pumps, gluconeogenesis, ureagenesis, etc., etc. A proportion is uncoupled towards heat generation. Depending on the organ, muscles can generate more heat and also reactive oxygen species. And non-mitochondrial processes are used by about 5% of total oxygen consumption. New mitochondria can be regenerated by a process called biogenesis, and old damaged ones removed by mitophagy. So mitochondria are broken apart, fission, and the damaged mitochondria can be removed by mitophagy and then swallowed up by the autophagosome. On the other hand, the healthy bits of mitochondria can be fused together to form new functioning mitochondria. So there's a very regular, rapid turnover of new healthy mitochondria. The mitophagy is important for getting rid of the old damaged ones. To give a quick example of how hormones are made by mitochondria, cholesterol is brought into mitochondria through STAR, the regulatory protein. It's converted to pregnenolone. Pregnenolone can be used for lots of other hormones, sex hormones, aldosterone, but it can also go via 3 steps in the endoplasmic reticulum back into the mitochondria, and the 11 beta hydroxylase converts it into cortisol within the mitochondrion. Tyrosine is converted to DOPA by an enzyme, tyrosine hydroxylase, situated on the mitochondria. That's converted to dopamine, norepinephrine, and then epinephrine, and they're broken down by monoamine oxidases that again are tethered to the mitochondria. So in summary, mitochondrial respiration provides the necessary ATP to fuel complex metabolic processes, but there are many, many other roles that the mitochondria plays. Oxygen delivery is dictated by metabolic demand, which tells the mitochondria work harder or work less hard. As I mentioned before, not all oxygen use is channeled towards ATP production, and the mitochondria is important also for generating reactive oxygen species, as well as heat. Thank you very much indeed for listening.